Hey guys, this is Chris back again with the Ancient Scholar, and I'm going to talk about the last major substance or molecule in the serotonergic psychedelic substances, and that is a molecule of mescaline. Mescaline is common, and it's somewhat popular here in the United States. We see it used by the Aboriginal people in ceremonies that have a very spiritual or religious nature to them. And so we see it used here in the United States by the Aboriginals in that sort of medicinal, spiritual, religious context, just like you see things like ayahuasca used in Brazil and Peru. There is some synonymous um, associations that you can make between the two situations because they're very similar. Um, so this is a molecule of mescaline. Mescaline comes from certain species of cacti that are relatively common in the Southwest, where I happen to live. So when we look at the molecule of mescaline here, it does kind of have, let's hold it up like this, it does kind of have the backbone. Okay, it does have the biogenic amine backbone. You still have your aromatic ring, and you have your amine side chain here topped with a nitrogen. However, what makes this really different are you have these three oxygens that are covalently bound to the aromatic ring, and then you have methyl groups topping those oxygens there. I should say that this is not a ring here. Okay, these methyl groups are separate. It's just kind of how the way the hydrogens are kind of touching. So maybe I'll pull them away from each other just a little bit, just so there's no confusion as to what's going on there. That this is not a ring. That these methyl groups come off separately. So there you go. This is molecule of mescaline. It still has the very similar. It has a similar structure to all the other biogenic amines, with the exception of these methyl groups here. So it still has pharmacologically. It's still going to have a serotonergic effects, like many of the other serotonergic agents. Okay, so that's really all I have to say about the molecular structures of the agents. Hopefully you found these series of videos helpful. I should say that I spent a lot of time on the serotonergic agents because they're so common, they're so popular, and there's a lot of data and a lot of popularized stuff out there on them. So it's always nice to kind of inject a little bit of the science and talk a little bit about what we know is going on. What we know is going on is actually very complicated, and even though we call these serotonergics, it should be noted that there are multiple neurotransmitter systems involved: dopamine, glutamate, other receptors. I mean, there are over a dozen subtypes of serotonin or 5-HT receptors. And like I've said in other videos, some receptors are agonized, some receptors are antagonized. And another thing that I just want to kind of say here, and I'm guilty of doing this as well, is that we sometimes tend to oversimplify the receptor model of pharmacology. It's such an important model. That's how a majority of our agents work, is they interact with receptors, not only just psychedelics, but I'm talking about pharmacology in general. And we sometimes tend to oversimplify it when we're teaching people about pharmacology, and we say that it's like a lock and key mechanism where the key goes into the lock and you open the lock and you close the lock. That's not entirely true at all, actually. It's not. There's very little truth to that at all. It's just that particular model works good in some situations, like if I give somebody albuterol, I can say that it interacts, it agonizes beta receptors. The beta receptors are G protein linked, G proteins, adenyl cyclase, cyclic adenosine monophosphate, or 3' 5' adenosine monophosphate, and eventually you have bronchodilation, and that's a good model. And you don't necessarily need to go beyond that model to have a basic understanding for how something like albuterol would help somebody with asthma, say. But once we get into the psychedelics and other types of pharmacology, there are some very important things that come out with receptors, and that is that when we talk about a molecule, okay, we talk about a molecule interacting with a receptor, um, it's not a unlock lock kind of situation. The receptors are, particularly the serotonin receptors, are what we call G protein coupled, which means that they are a member of the TM7 a family, superfamily of receptors, and TM7 stands for transmembrane 7, and the receptors actually have 7 little coiled parts that penetrate the cell membrane, and these little coiled parts all kind of come together to make up the receptor. And what happens is when a ligand comes in and interacts with the receptor has what's called a conformation, and that conformation changes slightly. And it's that conformational change that then acts upon G proteins and other intermediate signaling systems within the cell. So just like I have my glasses here, maybe this is one conformation and this is another conformation, but could I not have maybe a conformation like this, or a conformation like this, or like this, or like this, or like this? Those are all different conformations of my glasses, and just like I can have several conformations here, these receptors can have different conformations. So it's not necessarily just a lock-unlock, it is multiple different conformations, and each conformation may have a slightly different effect on different signaling systems within the cells. And then you have to take into consideration that you have millions and millions of different neurons going to very different areas of the brains, and some of them are agonized, some of them antagonized, and some of them have this conformation, some of that conformation, and it's the sum of all of these effects that really creates, consciousness. So when we alter consciousness, this overly simplistic view of, oh, a receptor is agonized or antagonized doesn't lead us to any sort of good understanding of how these substances work. So we're finding out that really there are individual little conformational changes that occur, and there can be several conformations and several different effects that can happen on intracellular signaling. So that's certainly something that we want to take into consideration as we think about the complicated effects of the psychedelics, and as we think, when we compare LSD to DMT to mescaline to psilocybin, they all generally work on serotonin and serotonin receptors, but we know that in those individual meds or drugs can cause very different signs and symptoms, even though they are considered, hallucinogenic, quote unquote. Even the terminology that people use is kind of all over the place, and some people may dislike my terminology and others may like it. I use that in a very broad kind of term. Some people even prefer the term entactogens, which are substances that have some stimulating effects, but psychotropic. There are lots and lots of different terms that you can throw out there, but I just kind of wanted to emphasize that there's a level of complexity to this that needs to be appreciated if you're really able to step beyond just a basic understanding that often people like me, unfortunately, resort to when people are first learning pharmacology. Okay, guys, I hope you've enjoyed this series of videos. There'll be more to come. As always, thanks for hanging in there.